Cloning organisms. Cloning organisms is a little bit different from the gene cloning that we looked at before. With gene cloning, you just take one piece of DNA for one gene and try to get that into a plasmid and then into a bacterial cell so that you can copy lots of copies of that gene. In organism cloning, you're cloning the entire animal or the entire plant or the entire organism. And so the offspring that's made should be genetically identical to the parent. It's very much like asexual reproduction. So, why would you want to do this? One reason would be to produce animal models of the disease. So, let's say you're studying the effects of a certain drug on heart disease in mice, and you're just starting these trials, and you want to take out the as much variation as you can to study the genetics. So, you might actually want to study uh, mice with all the same genetics, or maybe you're interested in mice that have a certain genetic problem so that you can treat people who have a very similar genetic problem. You might have one mouse that has this problem, and if you clone that mouse, then you'll have plenty of them for your research with to see if this drug works well with them. You could also do this to get stem cells that you might need for research, or you might want to produce more of a certain transgenic animal. So, what is a transgenic animal? It might be a goat with a piece of DNA from a different organism. So, the trans means from a different organism. So, it could be from a human. So, it might be a human uh, hormone for, or a human gene for growth hormone. And then you get that into the embryo um, of a goat and then implant that in a goat, and you have this baby transgenic animal, and it's able to produce, let's say it's a female goat, and it's able to produce milk that has this growth hormone that you're interested in, and then you can just isolate it from there and give it to people. But you've only got one goat. So the cloning of that organism would be able to get you more goats that could do the same thing. So with the early studies, they would take an organism like maybe a tadpole, like a baby frog, and take the nucleus from an intestinal cell, put that into an embryonic cell and see if it would grow. So here's what they did at first. They would take a frog egg cell, that's this, and they would denucleate it or enucleate it. That means you've got the cell here with no nucleus, so no genetic material. And if they have a frog embryo, here it is at only the 4-cell stage, the differentiation that happens between that cell and that cell and that cell and that cell is very minimal at this point. So they're going to take one of these cells and they'll take the nucleus from it and they'll put it inside this enucleated egg. And so now you've got the cytoplasm of a brand new egg, but you've got the nucleus from a 4-cell stage. That will typically grow up into a tadpole and be a very normal, happy tadpole. If instead you take a tadpole that's older than this 4-cell stage, one that's already differentiated, maybe it's like 3 or 4 days old, if you take one of the intestinal cells and take the nucleus from it and put it into this egg cell with no nucleus, they can grow up that zygote into an embryo. And the problem was that most of the embryos would actually stop developing before they got to the tadpole stage. So scientists have been working on this for quite a long time. And in 1997, the first scientist was successful with this. They used a sheep named Dolly, and the baby, I guess, was Dolly, and it was cloned from an adult sheep, and it was born viable, and yay! Except the problem was that the sheep died early, actually. So let me go to this part. So Dolly died early, and she had arthritis, and they think that maybe it's because there were some changes that happened to the cell as it developed, right? So if you take a cell from an adult sheep and take the nucleus and put it into an egg, it's still, even though they were able to get this sheep to be born, it wasn't completely reprogrammed to be a normal little embryonic cell. So here's what they did. First of all, they have an egg cell donor and they take out the nucleus. So that means you don't have the genetic material of this sheep, well, except the mitochondria, right? But anyway, they took the nucleus out, and so here's the just the plain old egg cell with no nucleus. Whoops, no nucleus. And then they take this donor sheep, and I don't know why they used memory cells, but whatever. They took some cells out and they took the nucleus. So that means this nucleus nucleus had the DNA from this sheep. So I'm going to call this sheep number 1, and this will be sheep number 2. So now they're putting together the nucleus from the sheep cell and from the sheep, and the egg cell from this one without the nucleus. And there you have your zygote. And so there was no fertilization. It took an adult nucleus. And then it grew into an embryo, and they took that embryo and they implanted it into a surrogate mother. So they put it into her uterus. This is sheep number 3. And then you have the baby sheep born. And so that was the first successful cloning experiment. And so, what I would want you to know on AP is that this sheep would be genetically identical to sheep 1 or 2 or 3. And so, the answer should be sheep 1 because they used her nucleus from her egg cells. They the sheep wouldn't be identical to donor number 2 except for the mitochondria, and wouldn't be similar to the sheep number 3. It just grew in sheep number 3. Okay, so most nuclear transplantation studies, most of the time they don't really work great. Usually the babies have some kind of problem, and the reason is because there are lots of changes that happen to the DNA over time, right? So as the cells do mitosis, they also differentiate, and to differentiate, they turn on some genes and off other genes. So the changes that happen to the DNA include, and to the histones too, right, include acetylation of histones or methylation of DNA or of histones and phosphorylation and all that stuff we talked about a couple of units ago. So all of that stuff has to be removed in the nucleus from the donor animal in order to express the genes appropriately, in order to take all that stuff off so you have a brand new baby cell or baby DNA. And that's really difficult to do. So that poses a pretty big problem. So now I have a few definitions that I want to go over. First of all, a stem cell. So a stem cell is an unspecialized cell that can reproduce itself and differentiate into other types of cells. So if you take embryonic stem cells and you take one of these cells and you take it out of the embryo and you grow it up, these cells should become any other type of cell. So these embryonic stem cells, we're going to call those pluripotent stem cells. If you have an early zygote, like let's say you have a zygote right here, that zygote is something we call potent, potent. And you don't really have to memorize these terms, but just the concept that this can become anything, can become any of the cells. It can become the placenta, it can become any of the cells of the body. Once you get to the stage where you have an inner cell mass growing, these cells are what we would call pluripotent. So P-U-R-I, pluripotent. So these cells from the inner cell mass, which is this part right here, can't become placenta and stuff, but they can become anything in the body. So liver cells, nerve cells, blood cells, et cetera, et cetera, et cetera. And then we have these cells called adult stem cells. These are multipotent, meaning, whoops, supposed to be potent. So that means these cells can become lots of other cells, but not any other type of cell. So these cells can become many blood cells. They can become erythrocytes, the red blood cells. They can become any of the white blood cells. There are a whole bunch of them. Monocytes, any of the white blood cells. So multipotent means this is what adult stem cells are. They can become many types of cells, but they're already partially differentiated. So these stem cells shouldn't be able to become liver cells or nerve cells or the others. So stem cells isolated from early embryos are called embryonic stem cells, and that's really what researchers want to study most of the time because they can become any other type of cell. Researchers now have been able to take fat cells or skin cells from people and bring them all the way back to this embryonic stem cell stage, which makes them pluripotent, which is super, super useful to use. Back not so many years ago, scientists couldn't do this, and so they were researching with embryonic stem cells, and there was a lot of controversy there because a lot of people were very uncomfortable with scientists using human embryos. But now there's really not that problem anymore because we can take these cells from adult cells and bring them backwards toward early embryonic stages. Okay, so this part, the adult body has adult stem cells, which is what we're talking about here, which can replace non-producing specialized cells like blood cells, for example. Okay, so this is what I was just saying. Researchers can transform skin cells, fat cells they've used too, into embryonic stem cells by using certain viruses to introduce stem master cell regulatory genes to backwards, almost backwards in time to reprogram the cells to be like sort of baby cells. So these are called induced pluripotent stem cells because they can become any of the type of body cells, but induced because they weren't always embryonic, right? They had been adult stem cells for a while. And so, what you might do, the research here is kind of amazing. Research from this particular, so skin from this particular patient, you reprogram them, make them into induced potent stem cells, and then treat them so that they're going to differentiate into another stem cell, another differentiated type cell, and then return them back into the patient. So what you can do with this is really amazing. They've been able to get mice with broken spinal cords able to walk again, which is mind blowing, right? They could take these and actually genetically engineer them at this point, right? While they're still stem cells. Um, and maybe re-engineer them so that they don't have some kind of mutation. Maybe it's for sickle cell anemia, right? And put that back into your bone marrow so that you don't reject them because they're your cells. Or maybe treat them so that they are white blood cells that will attack a cancer that's in your body. So there's just a lot of really, really amazing, fantastic research that's going on in this area right now. So these cells can be used to treat some diseases or replace non-functional tissues like a broken spinal cord. We're not at the point yet where we can do that in people, but there's been success with animal models.